So first, I'd just like to say that I really enjoyed reading your thesis. That I guess there's three things that I just like to point out that I appreciated. One is the learning about the evolutionary significance of lipids in the adaptation of Calanus finmarchicus especially. That I thought that was very, very interesting. And the use of that for understanding and predicting how Calanus and other copepods might respond to climate change. Mm-hmm. We'll talk a little bit about that, I think, as we go on. But those two things in particular I found very, very useful and interesting. And then finally, I found very nice your philosophy of science. And as you know, many of the people that you acknowledged in the introduction to your thesis, I think I know many of those people well. Cindy Lee, who you acknowledged, cut my hair in graduate school, as an example, at Scripps. But Carl Bonza was my professor at the University of Washington. And you made one statement in there that I have tried to keep it honest. And I think that is really a very important statement that I think I and other people can trust your results and the honesty that they represent. So I'd like just to compliment you, first of all, in those three areas and say that I enjoyed reading the thesis. So what I'm going to do is I have a list of questions on my iPad here, which I'll work off of. It's the easiest way for me. And the first one involves the differences between taxa of phytoplankton and their lipid content. Mm-hmm. I'm not a lipid chemist, but nevertheless, I appreciated in reading your thesis that taxa of phytoplankton differ, groups of phytoplankton, if you will. And as an example, cyanobacteria appear to lack triacylglycerides tags. And my question, and really it's something that you talked about, but I'd enjoy having you talk more about it and perhaps talk about the future, is what differences in the composition of the phytoplankton make in terms of their nutritional usefulness to copepods grazing on them. All copepods aren't equal. And as an example, during succession, often, Margulis mandala, we start with diatoms, and then as the nutrients are taken out of the water and it warms up, we end up with dinoflagellates quite often. And as you move from near shore, you move from a diatom-dominated assemblage quite often to one that's dominated by picoplankton, cyanobacteria. Mm-hmm. What are the effects of these changes in time and in space on the suitability of the phytoplankton assemblage as food, and in particular, the nutrition for lipids in copepods?
Well, as I kind of mentioned before, if we start with the diversity of fatty acids in the different algal taxa groups, it's quite interesting that they have a different kind of a profile or different kind of a fingerprint. These different, it is kind of genera-based. And within each each group, there is also some kind of variability, but generally they are fairly similar. So that kind of leads to the conclusion that must be evolutionary, some kind of reason for that. And they are dealing with kind of, and there's two things. Either they are dealing with the same problem in different ways, just the variability, how we deal with our environment, or they are optimizing their physiological functions. So, like we were talking about with the Chekley here before, that was is it the light? Is it a nutrient? Is it that affects, that decides, that determines where the fatty acid desaturation goes, or how long the elongation of fatty acids go, for example. It makes a huge difference for the consumer, how far they go. They like I say, the phytoplankton cell is not doing it to be a good food. There would be they counterbalance their goodness by making to be toxic or small or too big or whatever to consume. So as a contraction is a combination then of these factors what makes the goodness of the cell as food for the next level. Copepods, the density of copepods is highest along the in the shallow and the shelf seas, for example, and where the phytoplankton cells are larger, and they, I think also it's all kind of based on evolutionary trends, where they have found their optimum. But what is happening quite often now, for example, with change in our environment, change in temperature due to different reasons, is that we are throwing it a little bit out of balance. So how the copepods are, for example, going to change their adaptation to a different phytoplankton blooms is, there could be a little bit of a mismatch there, if it's correct to say there.
So as an example, as the climate of the Earth changes with warming and stratification and different nutrient availability with climate change, and the phytoplankton assemblage reacts to that in terms of changing its composition and perhaps even the chemical composition of the individual taxa, do you think that will have an effect on the lipid utilizing copepods such as Calanus finmarchicus?
Could certainly do, but we have to also look at the window when they are making the lipids. Most of the production of eggs and the growth stage is during maybe the diatom phase, when there are lots of these essential EPAs in diatoms. But I think the period when they accumulate in lipids, they are more heterotrophic. They are using the dinoflagellates-based diet and the heterotrophic-based diet more. I have not seen any studies doing this, to figure out, what on this how does the different phytoplankton affect the different kind of growth stage of the same population. Mm-hmm. But I think it could be quite important to know what type of food web they are basing the growth. And with warming climate, if there's going to be more stratification, there's going to be less nutrients coming up to the into the photic zone, for example. It's going to affect the composition of the phytoplankton there also. So yeah, I believe there's going to be some effect, but how drastic it is, it depends on the availability, if it affects the concentration of the algal of the phytoplankton, the production of them. So there's a lot of ifs and buts and what ifs there, like everything with global climate change there. Yeah. Okay.
Yeah. Another area that I found interesting was the use of the lipid composition of phytoplankton as trophic markers.
Yeah.
And I think you called them fatty acid trophic markers, or FATMs.
Yeah.
And the idea, I think, is that in the plankton, it's very difficult to know what consumers are eating because we can't observe that directly, generally. And therefore, if you could look in their guts or in their bodies in their entirety and identify compounds that are specific to certain food types, you can infer about what they've been eating. You mentioned also that you can use that to determine the trophic level, whether they're primary consumers of phytoplankton or whether they're primary consumers of heterotrophs or secondary consumers. And can you comment a little bit about how that might, and maybe I misunderstood that, but could you comment about that a little bit, the utility of trophic markers to infer about both what's being eaten, who eats who, and also about the trophic level? Please.
Copepods, they kind of, there's in my thesis, I summarized these studies, and I have a slight skepticism towards these utilization of these, because there is a preferential accumulation of certain fatty acids, for example. So they are going to be skewing a little bit the uses of certain taxa of phytoplankton. I think there's actually the best use of fatty acid, the tracers, fatty acid tracers, is exactly to determine the trophic level, because there are certain fatty acids that that are very what can I say? There are certain fatty acids that are the Copepod make it themselves, basically. And so they have to transfer the fatty acids somehow, and they have to they can utilize them and they also utilize them preferentially. So it's, but there are certain very conservative fatty acids that we can use, and they can tell you, for example, if they are based on diatoms versus the flagellates. But you cannot really go very much more specific into that. If you go into higher trophic levels, for example, with fish and fish larvae, there's a certain fatty acid tracers that are typical for certain copepods. So we can tell them, we can read out of their profiles if they have been eating calanus, for example, and what kind of calanus they have been eating, because of the specificity of them making certain fatty acids. And there, I think, it becomes important to be able to say if they are based on, if they have been eating calanus or not.
Thank you. Okay.
The use of fatty acid trophic markers, I am not totally sold on the idea. It works very well when you have a clean laboratory experiments, but it's a little bit more fuzzy in nature, I have to say.
Thank you. That clarifies.
Mm-hmm.
An area that wasn't mentioned in the thesis that I would enjoy your comment on is the topic of high nutrient, low chlorophyll areas, or HNLC areas in the ocean. So many of the areas of the ocean, primary production, or the ocean has lots of nutrients, nitrate, and silicate, and phosphate, even towards the end of the growing season. And this is because iron limits production there, the micronutrient iron. And these are HNLC areas, and notably the North Pacific and the Southern Ocean are such areas, as opposed to the North Atlantic, which is not an HNLC area. And you mentioned in your thesis that iron, when it's limiting, affects the lipid composition of the phytoplankton. And I'm just curious whether or not the phytoplankton in the HNLC areas, in your opinion, might be affected by this lack of iron in the environment, and therefore there might be a different lipid dynamic, if you will, in the North Pacific and in the Southern Ocean as opposed to the North Atlantic. It's very speculative, but I enjoy if you understand, if you gather what I'm trying to ask.
Basically, the iron is very important in photosynthesis.
Yeah.
You know, there's one of the most. It gives electron to the activator, the photosynthetic thing. So I think there is the limitation, is the photosynthetic activity. Will be. Therefore, when you're adding iron, you kick off the productions in the phytoplankton cell, and therefore the fatty acid production. I think their iron becomes important, just in starting the photosynthesis. Depending on what algae are going to take over, that are usually picoalgae and stuff that take over, aren't they? In these
they're smaller forms,
very smaller forms. It depends what takes over, how they're going to act. But I think it's a photosynthetic limitation.
Do you know if this brings me to another question, and that one of the more fascinating parts of the thesis, I thought, and that is the difference between the Arctic and the Antarctic.
Yeah.
And maybe you could tell us a little bit about the differences in the lipids that are formed by the copepods in the Arctic and the Antarctic, and that relationship to the physical water column. And
the copepods that I've been studying are in the northern Arctic waters, polar areas. They mainly make these Worcester lipids. While in the southern oceans, they are copepods with also accumulated huge amounts of lipids, but they are the triacyl glyceride lipids. So these are the guys with 3 fatty acids versus one fatty acid, 1 alcohol. And this is also kind of just an observation and speculation why this is. Why do they accumulate so different kinds of lipids? And the Worcester lipids has been associated with this deep diapause, while the triacyl glycerides are more active. The animals are more active and survive, are active over the winter time. This has probably to do with the depth of the mixed layer in the Antarctic. There's a much more mixed water. The mixed layer depth is very deep. And so they don't find any quiescent areas to go into diapause. So they have to adopt a different strategy to deal with the shortness of the season. A second thing is that the shortness of the season in the Antarctic is not as short as in the North, because the land mass of the South Pole, it goes all the way to I think it's about 60 degrees south. Well, up in the Arctic, we're talking about the polar areas. That's the water all the way to the pole. So the Arctic, so the length of the day is quite different in those. So the growth season is also longer there. So they can deal with the period of famine is much shorter in this southern area. So combination of these two, they made the possibilities for the animals, both copepods and krill and other, to survive this period either by accumulating these large amounts of then triacyl glycerides that is much more accessible to use. You can burn them faster, you can utilize them more easily, and again then build them up a little bit faster. So there is a more kind of dynamic in the lipid utilization of these animals. So that is these two reasons for I think there's a major difference. There are areas though, there are copepods that do have a Worcester-based diapause. And they are maybe more in the fjords and in the areas they can find more quiescent water to enter these diapause.
So I'm curious, are there implications to the food webs in the Antarctic for the copepods having triacyl glycerides as opposed to wax esters? Are the crab-eater seals and the baleen whales, and this is once again very speculative, but would there be any reason for the consumers to be different in terms of their dietary needs or utilization of the plankton? Or if you wish not to comment, we'll move on. That's fine.
I'm not quite sure, but okay. They're more accessible. They're not as much out of the water, so I think there might be more richness of the year-round rich availability in the south. But in the quality, I don't think so, because these are also fatty acids based and.
Is there a broader range of consumers that's able to use tags as opposed to wax esters, or is it that not necessarily the case?
I don't know, but you need the specific, to be able to use the wax ester, you need the specific enzyme to be able to break the wax esters down. Yeah. And not all have them, and I think it would be interesting to see if the Southern Ocean species have that enzyme, so they just they don't have to develop it if they don't face it.
So Dick touched on this a little bit, but I'm sure you're aware of it. But there was a study of Inuits in Greenland recently published in Science that showed that the Greenland Inuits have evolved to genetically and physiologically be able to use omega-3 fatty acids, whereas the general population outside of Inuits is less able to do that.
Sorry, what do you mean by using them in
metabolize them.
Okay, yeah.
Yeah,
sorry.
and so that this may be getting off the subject a little bit, but it actually pertains to what you just talked about with the Southern Ocean and as opposed to the North Atlantic a little bit, that organisms have adapted even within a species such as humans to use omega-3s in different ways, in different amounts, different capabilities. And so one of the observations is that omega-3s may not be as well utilized by non-Inuits as by Inuits. And it would be comparable to some taxa in the Southern Ocean, let's say, being able to use tags but not being able to use wax esters.
Yeah.
But, we'll stop there. Yeah. But this gets, I mean, to me this is a really interesting area. You talk about also in the thesis that some copepods, benthic copepods and copepods feeding on detritus, sinking detritus, may be able to use different components of the lipid assemblage than those feeding near the surface on diatoms, as an example, like Calanus finmarchicus. So would you expect Onsia or Oethona, both of which tend to feed on detritus, live on detritus quite often, to have different genetic capabilities for utilizing wax esters than copepods such as Calanus finmarchicus?
To utilize wax esters?
Or well, lipids in general.
Yeah, okay.
Perhaps.
No, to upgrade,
acids,
to upgrade basically the there's probably yes, I would, but the idea. This is kind of what I was collecting together, all the information. It started basically with the ciliates. There has been studies showing that the ciliates have been able to upgrade the quality of the algae. For example, a very omega-3 low chain omega-3 fatty acids up to EPA, so to a long chain usable high quality fatty acid. Then we did a study that I presented in the thesis that this was not always the case. And then when I was doing the summary, I was looking into, okay, who can do this? And there seems to be the copepods and particoids that can do it, so they are particle, and then probably Oethona, if it's living on totally on the, if they're totally living on detritus, and the ciliate that has been shown to change this. They're all benthic. They're all living on a very poor food source. And through some literature, I saw that there was kind of confirmed by some other studies that, or at least this hypothesis came up in other studies, that it is based on the low quality food they have to live on. They have to be able to build up the quality of they have this ability to elongate and desaturate some, mainly elongate the fascia. And that is also, it's kind of a cleaning up a little bit in the understanding in the literature how this works.
So what do you think the role of genetics is, genomics and transcriptomics, for elucidating some of these problems? In other words, looking for genes that regulate the digestion of different types of lipids and their presence in different species of copepods or other plankton. Do you think there's potential for doing that? Is that something that you feel has value? Or?
What I understand about, I wanted to do something about this, to use transcriptomics to understand enzymatic activities. And how I understood that, then it is kind of a, it gives you a kind of a very broad answer. You know if it's on or off, basically. If at least transcriptometry for enzyme activities, what I was looking for. For fatty acid elongation, I have to say I don't know enough about how this works, but I was a little bit disappointed by this to hear about it was just on and off kind of, and the person I was talking to, she said, why don't you just measure the enzymes instead of going into this complication of doing transcriptometry? And I think we are often looking for more fancy methods to use instead of using the more simple ways. I'm not really sure the enzymes, in my case, to use enzymes would be giving me the answer I want, but would need or like to know. But, and that's the same thing, I think, about the other. You have to be very specific in what you are, in your hypothesis building and what is the best solution to find that. And I'm not sure that genetics is always the answer.
Okay.
I don't think so.
Answer.
Yeah. They can tell you a lot, though. Yeah.
Okay.
I'm not
a geneticist,
no.
That's fine.
Yeah.
So another area that I found interesting was this one about the equilibrium at depth of Calanus finmarchicus at 600 or 800 meters, something like that. That it's if I understand correctly, an unstable equilibrium.
Yeah.
That if they go a little bit higher, they become positively buoyant and they'll shoot up to the surface like a diver.
Yeah.
Or if they go deep, they sink.
Yeah.
And I'm sort of curious what your thoughts are in terms of what sort of adaptations Calanus has made to avoid, to preventing loss from diapause, you might say, from the diapause depth. It seems to me that they have to act like robots in a sense. They have to sense their environment, and then they have to have an actuator that causes them to readjust their depth somehow.
Yeah, that's my ideas about it. They're using ionic exchange too. One of the most hypothesis I kind of mostly like is by David Pond. He is looking into how they use ammonia to use this balance. And to be able to excrete ammonia, you might have to burn up some protein or burn up some, and that could be a balance mechanism. You're losing your ballast by burning up protein, keeping your lipids. That's one idea. Another one, is this the ionic exchange with other, and that goes both ways. So they can maybe adjust it to that. Again, there's a problem with depth and pressure and everything. It's difficult to measure and do this in detail. Another thing was we were actually thinking about there might be, maybe they don't go exactly to the neutral buoyancy. If they stay a little bit above the neutral buoyancy and burn the lipids, they sink a little bit. So they kind of is this balance of being almost at your lipid buoyancy. I don't know. It's when we wrote this paper, we knew there was a flaw in it, and we pointed out there was a flaw, and there was a really fine flaw. I would say it was not a balanced model that we made. And of course, it was jumped on and caused a discussion, which is always nice. And but the thing is that it is a fine-tuning, and they have to deal with it because they are inactive. And they are inactive in this water. So how they do it, we are still, lots of nice hypotheses would be good to try to figure out, and nice technology to be able to do some experiments at 1,000 meters depth in the oceans. That's a dream. That might come through at some, of course.
And I'm not going to dwell on this, but Dick asked about it a little bit, and you mentioned it as well, Sigrún. In the past 5 years or so, there have been reanalyses of the abundance of mesopelagic fish. That using small trawls, they've been underestimated in terms of their abundance.
Hugely,
and using acoustics and larger trawls, now the abundance estimate has been increased by about an order of magnitude. And it seems to me that there is real potential for large mortality of diapausing copepods. Do you know, are there lots of mesopelagic fish, for instance, in the region where Calanus finmarchicus lives? Is that known? Or?
My educated guess would be yes, but there's again, they are deep, they are difficult to measure, and we haven't measured them. There's no measurement really of the population density in the North Atlantic, and I would think that would be the major mortality by mesopelagic fish. And interesting, I didn't talk about it in my talk here before, but when we were talking about the advective loss of calanus, I'd be really interested to see if there's a school of mesopelagic fish on the other side and see, waiting for this, 80 kilotons of calanus to come over the sill there, to see that is definitely a source of food for them. But of course, it's better to be in the layer and chop away. But we are talking about one of the most successful copepod species in the North Atlantic. So they have accounted for that mortality in the strategy, in the reproductive strategy and the overwintering strategy to try to maintain their population size, to be able to maintain the population.
My understanding also is that Norwegian herring in the winter dive and sometimes feed on diapausing calanus. If you can correct me if I'm wrong, but
I don't think.
And I've heard stories of them being brought up on deck and their stomachs exploding due to the high lipid content. And you've not heard of that, I guess.
Not in diapause, but I know this is.
I see. Okay.
And I haven't heard about it that they do it in diapause. Does herring go down to 1,600 meters? I don't know. I don't know. So somebody could answer that, Brian. No. But the thing is that yes, herring do Calanus phimicicus and Calanus is the major food for herring, and that is actually a huge problem to have Calanus in the stomach of herring, because herring is utilized. It cannot be utilized for human consumption if there's a Calanus in the stomach, because it ruins the meat. So all the herring goes into fish meal and oil. So it's a question of human consumption. It's very usable for others. And but that is. But I don't know if it happens during diapause. I think they're too deep.
It may not. I don't know.
Yeah.
The PNAS paper that was recently published, the last paper in your thesis, I believe, or following your thesis, dealt with the lipid pump. Yeah. And it's fascinating, and it's very important. There's also the pump that's due to migrating fish, mesopelagic fish in general, consuming near the surface and then going to depth and excreting. And we just talked about the importance of grazing by those animals on calanus at depth as well. The question I have, and maybe you could, you've explained that actually in your talk, is the relative low rates of metabolism and loss of lipid that you observed in the eastern part of the Norwegian Sea, as opposed to the relatively high rates that were in the model that you used of 44 to 92% loss at depth. It seemed to me that there was some discrepancy there, and I'm curious what your take is on that.
I don't think it's a huge discrepancy in it, but for example, in the Norwegian Sea, in the cold water, the carbon or sequestration carbon signal is based on the huge amounts of copepods over a long period of time, but they are respiring much less than, for example, in the Labrador Sea, where they're in the 4-degree water. And there's actually reported a huge lipid loss in those waters. And so the high percent is in the high. There's a smaller population with a big respiration rate and the higher temperatures, while the huge populations with lower respiration rates, losing less lipids, are in the eastern part of the Norwegian Sea. Also, when in my all measurements I have a significant error on them, and the measures are made on many copepods in this area, but there is always an error in the measurement. So the 44% loss might be within the error. You know, you could see that within the error of the. I haven't compared it really, but it doesn't. And one more thing, in this mass paper, we use a very conservative lipid volume, yeah. Because we didn't want to overestimate it, and so we used the medium value for that size copepod instead of, if you know, they are probably going for the upper values. So we are also overestimating the loss, percent loss. Mm-hmm. So that is my response to that one.
I mean, when you combine the lipid pump with the fish migration, along with the passive flux of particles, and compare those two former categories, the active migration and the lipid pump, are equal to or even greater than, it seems to me, the passive flux. But they're comparable, let's say. And so it's a very important finding. Very important finding.
We were very excited about it.
And as you say, I think the interesting thing in the future will be to go global and look to see how much that lipid pump affects global estimates of sequestration beneath the permanent thermocline. I've actually gone through all of the major points that I was going to go through, and so that I might rest my case now, if that's okay. Yeah, okay. Thank you, sir.
Thank you.
Very much. So we're getting closer. So I understand that the chairman of the committee would like maybe to ask a question or two. Is that still the case?
Yes, please.
Yes. Then please go ahead.
Okay. Yes, thank you very much, Sigrún, for a very interesting thesis, and I enjoyed reading it, and I've also enjoyed the discussion that we've had here this afternoon. I guess one of the things that I appreciated most was the fact that you were able to go from very detailed processes about the biochemical composition of phytoplankton through into the copepods, and to able to make a story all the way up to this last item that we were just talking about here, the lipid pump and the lipid shunt. And in your epilogue, you have a very nice paragraph in here, which I think you should read out. The last paragraph.
The last paragraph.
On page 86 of your epilogue about this. Some of the consequences the impact of this.
Shall I read it?
Yeah.
Okay. One of the research presented in this work underscores the is that one that you want, or the last one? Okay. Underscores the economic pressure involved. In our calculations of the Calanus microcube's advective loss over the Faroe-Bank channel in paper 19, we estimate that the annual 80 kilotons of carbon being flushed over to the Iceland basin and being potentially lost presents 400 million euros worth of omega-3 polyunsaturated fatty acids. In other words, in terms of weight, this advective loss is about a quarter of the current global demand for the omega-3 polyunsaturated fatty acids.
And I think you can read the last sentence as well.
While it's clear that there is a business interest, its potential would not have revealed if there had not been a curiosity-driven research in the first place.
Yes, thank you. I think that's a very good perspective on much of your work. That some of this work perhaps was not planned when you went out into the North Atlantic during the ICOS cruises with Mike Heath and Kathy Richardson and these people, that you would come up with these kind of estimates, but it actually came up as you were working with the data and digging into it and coming up with these calculations.
Yes, it's I think what I think has been kind of refreshing is that it is always evolving. You know, the ideas are evolving. You get new ones based on input from others. You know, it's always a flux between ideas from other people, and there you can build up some interesting hypotheses and test them and come up with and utilize your stone old data to try to figure out. You're really good of using stone old data, so I mean, and there is so much hidden there. And even though it was in the 90s, I mean, that's still valid and actually it comes up with some beautiful, yeah.
Yes.
Yeah.
Yes. Okay. I did have a couple questions about some of the climate change impacts. In your Article 16, where you compare the zooplankton ecology of Calanus finmarchicus and Calanus helgolandicus in the North Sea,
Yeah.
You demonstrated that in the summertime, in the stratified part of the North Sea,
Yeah.
they basically partition the habitat vertically, the Calanus finmarchicus in the deep, relatively cold layer, and the helgolandicus in the surface, warm layer. And you also have some implications in there about how you would actually sample those species and estimate their habitats based on the CPR studies. I was wondering, has anyone tried to actually map those habitats in a three-dimensional way, not only in two-dimension? I mean, it's very common to do habitat mapping in niche environment models in two-dimension, like as a flat surface. But here you have a clear case of a species having or occupying only part of the water column.
Yeah.
So,
has anybody tried? I think Mary Moore has, isn't she here? You know, I mean, there are some, but there are too few of these observations to be able to make a very good map of this. But now we can predict out from this finding that how the landscape is in these water masses. Most models are based on CPR data, and they are good in many ways, but they are, during this period of time, they're definitely not really good, because they do not see, there's a huge population down in depths that they cannot measure.
So what kind of data, or I mean, if you were to make a reproductive habitat, not a reproductive habitat, but a habitat of, say, Calanus finmarchicus in the North Sea in the summertime, what kind of data or instruments or technology would you need? I mean, if you were to make a grant to the EU or the Danish National Research Council,
send automatics, VPR sending all over the North Sea to get these separations. I mean,
but could you do it with VPR? I mean, could the VPR distinguish between those two species?
No, not between. No, they cannot.
Then you need, I don't know, direct ship sampling.
Yeah,
or,
just
more
life coat type research, cruises. I mean, we can I can endlessly say how important real truth data is, and correctly sampled real data is, and one of it is in the vertical, and you can just see that in the not just in the North Sea, but also in the North Atlantic. If they haven't been taking these vertical samples, there wouldn't be any, then we wouldn't know this much about Calanus finmarchicus as we do if we were been sampling in the upper 50 meters. So we have to go to 1,000 meters, 2,000 meters there. But in the North Sea, we have to know the structure because this is a crucially important species for the North Sea. And
yes, I mean,
and if you don't understand it,
and so on,
all the, for example, the sand eel is really important for the sand eel business, industry.
In sort of a climate change context, can you think of another species in the present North Sea zooplankton community that could perhaps take over the functional role of Calanus finmarchicus? Or are there species outside the North Sea, maybe in southern areas, from the species pool in the Bay of Biscay or I don't know where, that could perhaps
the Calanus helgolandicus is very similar to Calanus finmarchicus, and they are same size. Probably not as lipid rich, but they have the ability to build up lipids and have lipids, and but it's all about timing. The timing of when are they important for the fisheries, for example? When are they important for the growth of fish larvae, that are eating them? When is the timing of the production of these copepods? And what I think is happening with Calanus helgolandicus, it hasn't really matched yet the timing of the need in the springtime. They're still late. With the warming of the North Sea, they might be coming much earlier and be producing much earlier, and they might, the mismatch of that might be corrected in a way. That time we'll have to see. But my prediction, knowing these two species, I cannot really see a huge difference in them as quality food. It's a timing question of timing. When they are there in the right stages to be usable for growth, fish larva growth and the adult growth.
Okay. One last question. It sort of follows on from that. I mean, I know your sampling was sort of limited in the northerly direction around northern part of Iceland.
Okay, yeah.
But, I mean, in the Arctic Ocean itself, in the present day, do you know of any species that sort of also have the same functional role of Calanus finmarchicus, even further north?
Yeah, there are two other species that are, they always call the Calanus sister species and complex or whatever. As are the Glacialis and Hyperboreus. They are bigger. Calanus glacialis is more shell species, and Hyperboreus is a huge copepod with huge lipids, amount of lipids. But the abundance is often within the larger you are, the fewer you are. The abundance is not as high. So yeah, those are there, and equally, I think, important for the consumer fish consumers as finmarchicus is further south.
And maybe the lipid pump?
The lipid pump, yes. That is actually, we are working on...
Part of the going global?
And yeah, we go north. We're starting actually going north. We have actually, at the moment, a student looking into Hyperboreus, just a literature study and figuring out, And Calanus hyperboreus is a little more interesting, or different, interesting or not. It has a 3-year life cycle in diapause life cycle, and at depth, they broadcast their eggs at depth. So the female broadcasts the eggs at depth and dies. So there's an additional carbon loss there when the female is left behind, but the eggs go up, with the assistant buoyancy of lipids go up to the surface. So there's the same strategy for the 3 stages, for the previous stages, but the female, when it comes to the female stage, is going to be a different kind of lipid pump. It's not going to be as much lipid shunt by them, because the nitrogen is going to be in the female that dies and sinks. And but the eggs will go further up, and so that is a part of the little model that the student is going to make. Is to figure out this what a 3-year life cycle in diapause means, and taking into account the female mortality.
Okay, thank you very much.
Yeah.
I have no further questions.
Thank you. Then this will be the last time that I ask you if there are any questions from the auditorium. And if that is not the case, then we will move on. First, I would like to thank you all for coming, especially the opponents for coming such a far way. Your efforts are very important to quality assure our candidates, and I hope that after this event, you will be able to sign a little piece of paper, which is here in front of us, that says that this defense action has been carried out in a satisfactory manner, which is a crucial step in being awarded the Doctor Technicis. So with the paperwork in order, this will go to the Academic Council, which is actually meeting very soon, and the Council will then decide if you will be awarded the Doctor Technicis. But with everything in good order in terms of recommendations from assessment committee, usually that's a formality. But just to say, we can celebrate, but I mean, it ain't over till it's over, and the council always has the last word. This is a university. So with that said, I would again like to thank you all, and then I think having talked so much about lipids and fatty acids, maybe we should try some for ourselves. So therefore there is a little reception outside where you can all mingle and discuss this afternoon events while we just briefly meet in here to discuss whether we will sign the papers. And also there is a photo shoot that needs to be taken care of in here, so you should also stay. But the rest of you can leave the room, and we'll just briefly stay behind, but come out very soon. So thank you very much for coming.